Good afternoon. Good morning for those of you who join us from the other side of the ocean. My name is Carlos de Miguel, and I am the director of the Master in Biomedical Research held here at the University of Navarra. As most of you probably know, the main purpose of our degree, our program, is to instruct young, talented graduates in the field of biomedical research, so as that when they finish our program, they would be able to incorporate in a research team and carry on their own research project. Today, I would like to invite you and welcome you to this activity, which is part of our program, and it's called a masterclass. And today, we are lucky to have with us Professor Javier Novo. He's a professor of the faculty. He's a geneticist, and he is professor of human molecular genetics and computational biology in the degree of biochemistry at the Faculty of Science at our university. He is also an expert in the difficult field of bioinformatics and handling all these databases that are accessible now through internet to all of us. He has also developed an intense research activity studying chromosomal translocation in cancer, especially in hematological malignancies. And also he has focused in the analysis of genetic circuits that regulate the JAK-STAT signaling pathway, especially in myeloproliferative neoplasm. Actually, today this is the topic of his talk. His talk is titled Towards a microRNA-based Gene Regulatory Network of JAK-STAT Signaling. I want to thank Javier for his kindness in sharing with us his knowledge. And at the end of the talk, he will be glad, I'm sure, to answer any questions that you will like to ask him. I leave you with Javier, and I'm sure his talk will be of most interest of all of us. Thank you.
Thank you, Carlos, and thank you very much, all of you who are here and those who are over the internet watching this class for this master class for the Master in Biomedical Research. As Carlos said, I'm going to talk about some of the things we've been doing in the lab in the Department of Genetics over the last couple of years. The general topic, as you can see here, is towards a microRNA-based gene regulatory network of JAK-STAT signaling. And this is the building where we actually work here, the old research building of the University of Navarra, because there's a more new building which you might have heard of. Now, first thing I have to tell you is about hematological malignancies. In the Department of Genetics, for various reasons, we've had an interest in leukemia, in genetic and epigenetic alterations that lead to the development of hematological neoplasms, leukemia, etc. As Carlos mentioned, we have studied a lot of genetic alterations, that's basically translocations leading to these type of diseases. And over the years, we moved into this other field. So just a brief reminder of what hematological neoplasms are. This is a graph that depicts hemopoiesis. And you can see that from a stem cell, you can have common lymphoid progenitor cells and common myeloid progenitor cells, which then will give various types of committed progenitor cells that then will move on to differentiate as mature blood cells of the various types that we have. What usually happens in hematological malignancies is that one of these cells at different stages of maturation, of differentiation, acquires some changes, genetic or epigenetic, that lead to an increase in proliferation, a decrease in apoptosis, etc., whatever. something that makes that cell tumorigenic, and that gives rise to a clone, which then develops into the leukemia. Now, depending on which type of progenitor acquires that change, we can have lymphoid malignancies or myeloid malignancies, broadly speaking. And depending on the stage of maturation at which the genetic change takes place, we can have chronic neoplasms when the progenitor cell is more differentiated, or acute neoplasms when the progenitor cell is less differentiated, roughly speaking. Now, what we are going to talk today is about a type of myeloid malignancies, a subtype of this. So we are looking at changes that take place probably in some of these myeloid progenitor cells. And we are going to talk about chronic myeloproliferative neoplasms. That means that the genetic alteration takes place in more or less differentiated cells, which give rise either to erythrocytes, neutrophils, and platelets, basically. So it must be in a cell around here. Okay, so our interest in this field comes from, as I said, from various reasons, but basically these chronic myeloproliferative neoplasms, or MPNs, from now on, MPNs, are very famous in the field because of this particular type of myeloproliferative neoplasm, which is the chronic myeloid leukemia. The chronic myeloid leukemia is caused by the translocation between two chromosomes, chromosome 9 and chromosome 22, which gives rise to this derivative chromosome 22 known as Philadelphia chromosome. The outcome of this translocation is the generation of a fusion gene. There is a gene in chromosome 9 called ABL. This is a kinase. There is a gene in chromosome 22 called BCR. And the translocation, as you see, creates this fusion gene, which gives rise to a fusion transcript and gives rise to a fusion protein, which contains domains from the BCR gene, from the BCR protein, and domains from the ABL protein, which has, as you see, has a kinase domain, several SH domains, etc. Now, this fusion protein leads to constitutive kinase activity. ABL is a cytoplasmic kinase, so that leads to constitutive kinase activity in a progenitor cell, so that creates the cancer, the tumor, the leukemia. Now, this is the best known MPN, myeloproliferative neoplasm, but there are some others, a few others. Well, these are all the pathways that are deregulated by the fusion protein BCR-ABL and how it leads to cancer. Now, there are other, as I was saying, there are other MPNs, not so well known as chronic myeloid leukemia, but they became also very famous a few years ago because in 2005, a mutation was found in a gene, and that mutation was found in a high percentage of the patients with some types of MPNs. The gene is JAK2, and this is the protein encoded by JAK2 with all the domains. JAK2, as you see, is also a kinase. It has a kinase domain with a couple of phosphorylation sites. Then it has another another JAK homology domain, which has a pseudokinase activity, and in fact, it is an inhibitory domain. It inhibits the action of the kinase domain. Then there is an SH2 domain for binding to other proteins and a firm domain in the amino terminal. Now, the mutation found in virtually all patients with a type of MPN called polycythemia vera was the same. It was a V617F mutation, which led to the destruction of the pseudokinase domain, which being inhibitory on the kinase domain, that then led to an increased kinase activity in these cells. And as I say, this mutation was found in, well, some hematologists say that all patients with polycythemia vera, which is a type of MPN, and in about 50% of patients with essential thrombocythemia and primary myelofibrosis. These are two other MPNs, myeloproliferative neoplasms, of different types. Further studies in many labs around the world, including our lab, tried to find what was the prevalence of this mutation in different types of malignancies, in atypical MPNs like chronic myelomonocytic leukemia, hypereosinophilic syndrome, etc. So it's also found in other types of myeloid malignancies, but not in high numbers, not as high. It's not so striking as in polycythemia vera. Now, what this is telling us is that the deregulation of a signaling pathway, because JAK2, as you are probably aware, is part of a signaling pathway in the cell, which is called the JAK-STAT pathway. What this is telling us is that the alterations of this pathway are very important for the development of these disorders. And there might be many other mutations not yet found in other members of this pathway that could explain the missing 50% here, or all these patients. They might have other types of mutations. And that's what we've been doing over the last years. As I said, the JAK-STAT pathway is relatively simple. I don't know if I can say that because Carlos is an expert in signaling pathways, but there are many other pathways which are more complex. But this is relatively simple. There is a membrane receptor which is activated by ligand binding, and then there is a JAK protein which is there and is phosphorylated and then recruits a STAT protein which goes there, is phosphorylated, dimerizes, then moves to the nucleus and binds to some binding sequence in target genes. We have a more clear graph here. So you see a cytokine, usually this is the cytokine receptor with a JAK, and then ligand binding then activates, phosphorylates these JAKs, which then phosphorylate a STAT, which dimerizes, goes into the nucleus, and binds to specific sequences in the promoter of target genes. And so that activates transcription of those genes. Now, we can see here, we saw it here, but I think it's more clear here, that there are also negative regulators of this pathway. We have these SOX proteins, this PTP, these are phosphatases. We have these PS proteins which inhibit dimerization and binding of the dimer to the DNA, etc. And it's, as I said, I think it's a fairly simple pathway. It's rather well known. We know a lot about effectors, all these JAKs and SOX, and phosphatases, etc., the negative regulators as well. We know all the JAKs in the cell. We know all the STATs in the cell. We know which combinations of JAK and STAT take part in signaling due to different cytokines, etc. What we don't know so well is what genes are activated by binding of specific STAT dimers. And that was the area where we wanted to concentrate, to identify new target genes. Now, specifically of the JAK2 STAT5 signaling pathway, because JAK2 is the one which is mutated in MPNs, and we wanted to study myeloproliferative neoplasms, which did not have mutations in JAK2, the V617F mutation in JAK2. All those patients who do not have that mutation might have other mutations. So we focused on that, on those patients, and we wanted to see what was happening to the target genes, and if we could identify new novel target genes regulated by activated STATs. So these are the goals in general of our project: identification and characterization of novel STAT5 target genes in the cell, in a normal cell first, and see how then they are deregulated in MPN patients. And together with this, once we have the genes, and as you know, there are many emerging themes in gene regulation, and one of them is microRNAs, so new mechanisms of regulation of the JAK2 STAT5 signaling pathway based on microRNAs, which are becoming increasingly important in gene regulation. And microRNAs, you all know, are non-coding RNAs, short non-coding RNAs, 20 to 22 nucleotides in length, which bind to the 3' UTR sequences of genes, of target genes, and downregulate them either transcriptionally or post-transcriptionally by degradation of the messenger RNA or by translational repression. Okay, so what are our models? So we use cell lines, and then we move on to patient samples. I'm going to talk about the cell line models, basically. We have several cell lines which are either inducible with interleukin-3, which is one of the cytokines that activates JAK2 STAT5. So you can see which genes are upregulated after treatment with interleukin-3. And then we have several cell lines with mutations, usually come from patients with myeloproliferative neoplasms. This K562 is a good model for chronic myeloid leukemia because it has the Philadelphia chromosome, the BCR-ABL1 fusion. And then HEL and SET2 are two cell lines which they harbor this V617F mutation in JAK2, so they have increased JAK2 STAT5 signaling. And we can inhibit signaling in those cells with specific inhibitors for STAT5. So, well, to cut a long story short, selection of candidate target genes first. They should be genes which have in their promoter sequences a putative binding site for STAT5. Previous studies over several years have led to the identification of these sites, these motifs in DNA. They are called gas motifs, which, well, the best of, or the more specific, the most specific for STAT5 is this one, which you can see here. Then we searched all promoters in the human genome, all promoters for all genes in the human genome for the presence of those motifs. We used several databases and several algorithms, etc. That led to, we tried to rank them, and then that led to 64 genes, which we studied. And the way we studied these genes was, as I told you, using these cell line models. This is MO7E cells, where we can activate the pathway with interleukin-3. And there's an immunoblot for total JAK2, for total STAT5, and for phosphorylated JAK2 and phosphorylated STAT5. As you can see, treatment with interleukin-3 of these cells leads to phosphorylation of JAK2 and STAT5. So the pathway is activated. And then we can treat them as well with interleukin-3. And this STAT5 inhibitor is a commercial STAT5 inhibitor, which is, we are told, very specific for STAT5. You can see that it decreases STAT5 phosphorylation, and then you can check the expression of target genes, how they behave. You can see what happens when you treat the cells with interleukin-3, when you treat them with the inhibitor. If it's a bona fide target gene of the JAK2 STAT5 pathway, the expression of that gene should increase with interleukin-3 treatment, and it should go down with the addition of the inhibitor. And this is what happens to FISH and OSM, this oncostatin M. These are two already well-known targets of the JAK2 STAT5 pathway. And sure enough, they behave accordingly. And this is what we looked for in our candidate genes. PIM2 was one of the genes we found as a novel candidate. It turned out not to be too novel because there's another gene called PIM1, which is known to be a target, but that was good. Anyway, so we screened a lot of genes using this strategy, and then we used a second strategy, is to do a chip, a chromatin immunoprecipitation with an antibody anti-STAT5. I'm sure you all know about chip and chromatin immunoprecipitation. Basically, you cross-link the cells, the DNA and the proteins. So all proteins bound to the DNA become cross-linked, and then you immunoprecipitate with an antibody against one specific protein of interest, in this case STAT5. And in the immunoprecipitate, then you will have all the small fragments of DNA that were bound to that protein. Then you undo the cross-links and you release all those fragments, and you can see which genes were there. So we did this in cells that were not treated with interleukin-3 and in cells treated with interleukin-3, trying to find by quantitative PCR if the binding of STAT5 actually increased after interleukin-3. So that should be seen in the chip. That was another line of evidence that there was direct binding of STAT5 to the promoter of those genes. So we had two lines of evidence, this expression and binding of STAT5. And, well, briefly, this is fish. This is one of our positive controls because we know it is a target of STAT5. And it behaves very well. It goes up with interleukin-3, and when you treat it with interleukin-3 plus STAT5 inhibitor, it goes down again. And then this is the quantitative PCR for cells not treated with interleukin-3 and cells treated with interleukin-3. This is the chip result. Is the number of times that you have fold. This is 160-fold more binding of STAT5 to this particular gas site, this STAT5 binding site in the promoter of this gene. Actually, these are two binding sites in the promoter of this gene in cells treated with interleukin relative to cells untreated. The same happens to oncostatin M, another well-known target of STAT5. It goes up, goes down with inhibitor, and there is a gas site which is very clearly bound by STAT5. And then other genes that we found, PIM1, as I said, is already known. PIM2 is a novel gene because it's also, of course, very homologous to PIM1, but behaves pretty well. And then we found LIF, which is the one we liked the best because LIF is a very interesting cytokine with pleiotropic functions, and it's very important for the maintenance of the pluripotent state of stem cells. So it could actually, the LIF means leukemia inhibitory factor. So it's a good candidate gene for leukemia. And it behaves very well. It goes up a lot with interleukin-3, goes down with STAT5 inhibitor, and there is a gas site, a binding motif in its promoter that it's also bound by STAT5 according to the chip data. Then we tested this in the other cell lines, and well, this is another interesting result because these two cell lines, if you remember, they both have the V617F mutation that activates JAK2. So in theory, in these cells, the JAK2 STAT5 pathway is active, constitutively active, and the basal expression of LIF is much higher than in normal blood from a donor, and than in MO7E cells which are not treated with interleukin-3, so they should not have JAK2 STAT5 activation. And then when we treat these cells with a STAT5 inhibitor, again, you see, well, oncostatin M is our positive control. You see HEL. It it has the JAK2 mutation, so constitutive activation of STAT5. It goes down with the inhibitor, and SET2 cells goes down with the inhibitor. The same happens with LIF. So this confirms that it's behaving very well as a new target for the pathway. This is not the gene which we use. This is a STAT3 target gene, actually, because some of the referees were concerned that, referees are always concerned with something. So that we were actually activating STAT3, although we were using a STAT5 inhibitor. But this is STAT3, a known STAT3 target gene. So it shows that it's not behaving like STAT5 target genes. Then we moved on to patient samples. It's not easy to collect many patient samples for these diseases because there are not many in the world. Actually, we had to go to Galicia, to the other corner of the country, to collect these samples. That was a pleasure for me because I'm from there. So it allowed me to have some seafood. So if these are normal controls and these are primary myelofibrosis patients, these are polycythemia vera patients and essential thrombocythemia patients, in all of them, LIF expression was increased compared to normal samples. And it was very interesting that myelofibrosis patients were more increased than the other types of MPNs. There were only 3, because as I say, there are very few patients around the world with these disorders. Even so, that was significant compared to essential thrombocythemia, at least. But we are confident that if we could increase the numbers, this might be a very relevant result for myelofibrosis patients. Okay. Then I will talk briefly in the last part of my talk about the other thing I told you I was going to talk about, which is microRNAs. We were trying to find novel genes regulated by STAT5 and see how they behave in myeloproliferative neoplasms. And then I told you, and we also want to explore new ways of regulation, because a gene, nowadays in biology, we cannot do anymore the biology we used to do 10 years ago, which was this gene, this protein. You could study 2, 3, 5 at least. Now, biology is moving towards a systems level. So you have to study all genes and all proteins, or as many as you can, in order to have a more accurate picture of what is really going on in that cell. So there are many different types of regulation. You can study transcription factors and then genes and how those genes then inactivate those transcription factors, etc. And a very interesting way of trying to reconstruct these networks is using microRNAs, because microRNAs can regulate many different target genes. So what we did here was to use a platform. These tildes, these are Tachman low-density arrays. These are plates with a Tachman assay for each microRNA. Well, there are more microRNAs in humans than 644, but this was, well, what applied by systems had in this time. So you can screen the most abundant microRNAs. And then we did this in several situations. MO7E cells without interleukin-3, with interleukin-3, and with interleukin-3 plus STAT5 inhibitor. Again, we wanted to see which microRNAs went up by JAK2 STAT5 activation and then went down with STAT5 inhibition. We did the same for HEL cells and SET2 cells treated. Well, this is a JAK2 inhibitor. In fact, we also treated HEL cells with a STAT5 inhibitor and SET2 HELs with a STAT5 inhibitor. Remember, these two cell lines already have the activation of JAK-STAT because they have the mutation in JAK2, so we are inhibiting And then we are comparing both the situations, and so we can know which microRNAs go down in expression after inhibition of STAT5. And again, well, this gives you lots of numbers, and you have to try to refine your results and to come up with a set of microRNAs that behave coherently. So they go up with interleukin-3, they go down with the STAT5 inhibitor in MO70 cells, and go down with the STAT5 inhibitor in HEL cells and SET2 cells. When you have that small set of microRNAs, then you are quite confident that they are behaving properly. And these were these 9 microRNAs. But then we also took these 2. Basically, these 28 microRNAs were very interesting because they were behaving coherently in these 2 situations. Now, once you have that, you can start building up your networks. And briefly, these are, here the circles are the genes that we had found previously that were regulated by STAT5, by JAK2 STAT5. Red, upregulated, green, downregulated. And here in the center, these squares are the microRNAs, the 28 microRNAs that are, in this case, all of them are upregulated by STAT5. Now, the question is, which of these genes are actually targeted by which of these microRNAs? And then that's when you start having networks, and the thing becomes interesting. It's a bit of a nightmare, though, you know, but anyway. Because the problem here is that those are predictions. How do you know which microRNA targets which gene? Then you go to databases. But databases are predictions based on the binding site of the microRNA, the target site of the microRNA. They are not experimentally validated, all of them. So that gives you a bit of trouble, because then you have to validate everything. You have to try to filter and use several prediction databases, see which ones give you a good result in all of them or in most of them, etc. This was a lot of work. And then all these edges, these links, this means inhibits. The blue arrows means activates, because they are all of them activated by STAT5. But as you see, there are genes which are activated by STAT5 and are negatively regulated by microRNAs which are activated by STAT5. So this is where you have these regulatory loops. This, as I told you, is an emerging theme in biology. This is from Cell, one of the snapshots from Cell, the journal, where they were summarizing all the different types of feed-forward loops, negative feedback loops, integrated feed-forward loops, etc. How most processes in the cell can be explained by this type of circuits. You can explain many oscillatory patterns. You can explain steady state level, like expression of things, etc. So this is very interesting, but it's very computational, and it's usually only physicists like this, not biologists. So that's why you have to find a physicist or someone who is really interested in this type of thing. Other types of networks that you can build, for instance, these are negative regulators of the pathway. Remember PIAS, PTPN2. So some of these microRNAs actually targeted negative regulators of the pathway. So if microRNA is increased by STAT5, it will decrease the negative regulator of STAT5. So that will increase even further. See? So there are many potential ways in which this can be studied. And we are trying to do that just one by one. Or we have limited resources, human resources basically. So we are trying to select those targets which look more interesting. As I said, we are interested in LIF, and these are all the microRNAs that are upregulated after STAT5 JAK-STAT5 activation, and which target live. So we concentrated on this one because it was our best candidate, and we did some experiments in which you first have to find where are the targets for this microRNA in the 3' UTR, of LIF gene. You clone those regions in a luciferase vector so that you transfect, first you clone that region and you transfect some cells with the microRNA, with the pre-microRNA in the cells together with that vector. And then you should see first, when you transfect the cells just with the premier and a negative control premier, which doesn't bind, you should see that your gene of interest goes down after the cells, in this case health cells, are nucleoaffected with that premier, just that premier. You see here at different concentrations in this cell line. We also tested Z2 cells, I think. And this premier, the 3313P, which is the one we like best, behaved quite well in that it decreased the expression of endogenous LIF when you nucleoaffect those cells with that premier. And then we are now doing the luciferase assays in which you have to show specific binding of that premier to that target region in the 3' UTR of the LIF gene. Briefly, we did more studies with other microRNAs that we found that could be interesting. Some of them, I will tell you this very briefly, some of them, the genes targeted by these microRNAs formed a very interesting, a very compact network. These are proteins, actually, because it is from SMART, a protein database for interactions. And there are many oncogenes here. And in fact, when we mapped those genes onto the KEGG pathway for CML, for chronic myeloid leukemia, we saw they were enriched. All these green and red squares are genes targeted by one of those microRNAs. And it's nice that they are enriched in a pathway related to a myeloproliferative neoplasm, to chronic myeloid leukemia. And in fact, we found that in a cell line K562, these microRNAs are downregulated, and when you map them on with their targets, for instance, all these are the microRNAs which are downregulated in K562, some of their target genes are upregulated, and they are oncogenes, etc. So we are still trying to work out what's going on here. I mean, there's a lot of work to do. We're trying to concentrate on specific microRNA gene interactions and build little by little a wider picture in order to transfer these into patient samples. Once we have done all these studies with the cell lines, we will try to see what's the state of these microRNAs and these genes in samples from patients with myeloproliferative neoplasms. And this is a picture of the Department of Genetics, I think last year. So some of the people who've done this work, basically Elisa here, and Mikhail, he's not here, and Sarah here, and Inigo also contributed some work here. But well, most of them also contributed ideas, and Jose Luis, who's the boss. So thank you very much for your attention, and now I think I'll be taking questions either from the room here or from the internet, wherever you are. Thank you very much.
Okay, thank you very much for this wonderful presentation, Javier. And now it's time for, I think we have time for a couple of questions. Those of you who are connected by internet can make your question through the chat, and then we will forward them to Javier. And if anyone in the room wants to start with a question, you can ask now. If it's trouble for you or to ask the question in English, it's okay. You can ask it in Spanish, and then Javier will translate it.
I understand Spanish.
He will translate it in English, the questions, in case someone connected is not a Spanish-speaking person. So feel free.
Do we have anyone in the cloud? Or José Luis?
Anybody? Okay, I'll ask you one thing. It's a general question, because you have said that we have now these new tools that we can use to do a study of the genome-wide study, to look at a lot of genes at the same time. And you say this is good, because in the times before we were looking at 1, 2, 3 genes at a time. But at the end, you end up having to validate your results, and then you end up you have a lot of results, a lot of connections, very difficult to interpret, and then you have to go down to the experiments, gene by gene, and see this gene is actually behaving as the model is predicted by the model. How do you cope with that? How do you think we can cope with that?
It's terrible, isn't it? Because I wish I could do all the genes and all the interactions in the, but you see that's not feasible unless you are in a very big institute, the broad institute, or these big genome centers where they can do lots of stuff. So I think the approach should be, okay, you do the high throughput thing with the idea that that will enable you to identify things which otherwise you could not identify. If you just said at the beginning of the project, I'm going to study this gene, okay, that's okay, but that's based on your previous knowledge. This way you can first have a general picture, and then you can say, well, I like this, I like that, and probably you will discover things that you would not have discovered before. So I think that's the way we try to use it. Then you have to filter down your results and find the more likely candidates. If in a different situation, in a different setting, probably we could do more, try to validate all of them, but that's a lot of money and resources and people, and we are not such a large group for that.
So you would say that both approaches are complementary?
Yeah,
you need
then,
to start
as you
with
say,
that.
and then you finish with the same result, but probably you would not have got there if you had not done the...
Yeah, okay. Anybody? Yeah.
Well, maybe I lost the information, but I would like to know what kind of cytokines besides interleukin-3 interact with JAK systems.
Oh, well, there are GM-CSF, interleukin-6. Actually, well, there are many, but depending on the JAK and the STAT, because you can have JAK1, STAT3, and that responds to a cytokine. And but there are many interleukins, and GM-CSF, which is granulocyte macrophage colony stimulating factors. Many of these family of cytokines signal via JAK, different combinations of JAKs and STATs. In fact, well, and these receptors have two subunits, many of them, a common subunit and a different subunit, and some of those subunits, the genes, are also, in turn, are upregulated by some stats. So there are also feedback loops there. But yes, most of these family of interleukins signal via JAK stats. Yeah.
Okay, any other questions in the audience?
Oh, there are many mutations involved in all of these diseases, and my question is if there is one mutation or microRNA which could be good target for drug therapy?
Mm-hmm. Well, there aren't many actually, well, Professor Bitmanos could answer this because he's been years trying to find mutations, and there aren't many mutations apart from V61F, 617F, with not so prevalent. I mean, we found some new mutations, but in just 1-2 patients. So there might be many mutations, but very, very low prevalence, just almost patient-specific. And that's a problem because then you cannot create a drug that will target all patients. Yeah, this is a general theme, I think, in cancer research nowadays, when people are screening cancer genomes and they are finding that there are almost private mutations, that each cancer genome is different, although they all affect the same signaling pathways, but in different ways. So you might create a drug that targets that pathway, but it's more difficult. So in these diseases happens more or less that. We've been searching for mutations for quite some time now, for hidden translocations, hidden mutations, and that's probably not the best route nowadays. For CML, for chronic myeloid leukemia, due to the translocation and BCR-ABL1 fusion, that led to the development of a very specific drug, which actually targets the fusion protein, and that increased or improved the prognosis and the treatment of patients with CML. The idea is to do something similar for MPNs, to try to find mutations or ways in which these patients have alterations in the JAK-STAT signaling, so that we can create a drug to treat them all. If we could find the magic microRNA that controls everything, like the ring of the Lord of the Rings, that will control all genes and microRNAs, then that would be a good target. But it's not going to be easy.
Okay. Anybody else want to ask something to Javier? Anything from the chat? We have any questions?
From hyperspace or no, cyberspace or whatever it is. Yeah.
Well, I may ask another one because I have a lot of questions about this, but
you have 2 minutes.
2 minutes, okay. This is just another general question because I'm an expert. But this microRNA world seems to be something amazing now, as far as gene regulation is concerned. And the thing is that the other thing that worries me in this field is that you look for the sequences that might hybridate with the RNA and call them targets. But that's like you say, this is in silico experimentation. I mean, this is, and the thing is, which of those, because there are many. You've shown a picture here that in the same mRNA there are many targets, many sequences that can be targeted by one microRNA. And the thing is, which of those are real?
I wish I knew. But there are databases for validated targets, experimentally validated targets, but of course they are not all are there. And then these different algorithms for prediction use different, basically the seed, which is the 7 or 8 nucleotides, the strength of binding, etc. But they are predictions and they have to be validated, as you say. So in the end, we had 3 binding sites for LIF. In the 3' UTR of LIF, we had 3 potential ones, and we are trying to validate all of them. And again, that creates, one hopes that in 5-10 years' time, all targets will have been validated experimentally, and you will have a catalog, so you can go directly to the most interesting ones. But nowadays, you still have to do the experiments, yes. Yes, because predictions are not accurate enough yet.
Which is good, because otherwise physicists will take our work.
Yeah, we would be out of work.
Okay, I think I want to thank you again, Javier, for this wonderful talk. And now I just want to thank everybody in the name of the Faculty of Science, in the name of the Master of Biomedical Research at the University of Navarra for joining us to this activity, to this master class. And also those of you who have joined us from the other side of the screen, thank you very much for your attention. And please, if you have any question, any additional information that you may want to ask related to the master program, you know that you can easily reach us through our webpage. We will be more than glad to answer any question or any doubt that you may have about that. So if there are not any more questions, I want to thank you all, and thank you for your attention, and goodbye.